snowing up north.
Yeah,
So
we like went
no, like snow is stuck to the ground up north.
Okay. So, just a couple of quick reminders. Chapter 6 worksheet is due Wednesday. I did cut it down a little bit, so you should already be able to answer most of the first half if you looked at it. And then the last 3 or 4 questions we're going to cover today. I posted Chapter 5's worksheet answer key on MO2 this morning. So it is, it is just under the Dropbox for the homework 5. Okay, so there's an answer key. Alright. Exam is Wednesday. What's that? No lab. So I will be in tomorrow at normal time. Yeah. You can sleep in tomorrow. If you have extra questions or things you want to review sort of during that lab time would be a good time. I do have a meeting that is scheduled for 10, but if you go between 8 and 10, I'm happy to help you then. Or if you need to, tomorrow? Yeah. 4. Yeah. I have a meeting tomorrow that is going to be at least an hour, so probably 10 to 11. You can interrupt me during that meeting, it's just in my office, so if you that's the only time you can come see me, that's fine. And then I have a meeting at 3. What is that meeting? Oh, that meeting I won't leave my office. But otherwise, that was the other thing. I also posted a couple of extra practice problems. These are old labs, so you can obviously ignore the material that you don't need to focus on, but there are a couple of extra linkage problems in here. So this is basically a word question that walks you through it, and then there are a few additional mapping things that you can do if you want some extra practice. There's also a non-disjunction practice lab. This is what we're going to talk about today. This is going to be sort of the other big concept from this chapter. This is where you're going to have to draw out meiosis again. Okay, so we'll draw it out on the board again today, but again, a little bit of extra practice here. Okay, so for those of you who want some extra questions, this is a good place to go. Anything else? Questions? All right. So today in class we have 2 big concepts to finish up. The last part of what we started last week is translocations. So I'm going to just walk us through translocations. And then the final piece of this chapter I've shortened significantly from what I had intended to cover to just basically looking at non-disjunction. And so that is going to be where we have changes in full chromosome numbers as opposed to pieces of chromosomes. All right, so we talked about sort of this definition of translocations, right, where we have reciprocal and non-reciprocal, with reciprocal being 2 non-homologous chromosomes exchanging genetic information. Okay, so a piece of one breaks off and exchanges for a piece of another. We also have non-reciprocal, which is not shown up here, but that would be so if we have 2 non-homologous chromosomes, so we have A, B, C, D, E up here, and like L, M. Okay, non-homologous chromosomes. A non-reciprocal translocation would be something like reciprocal agreement. It means that both parties that are involved exchange goods or services. Right, so if it's reciprocal, both of our chromosomes exchange information. If it's non-reciprocal, one loses a piece that the other then sort of gains. All right, so this next slide gives us our definition and sort of runs us through the basics on reciprocal translocations. So again, in this situation, we are not losing or gaining any information. We still have the same exact amount of DNA in the same genes. What we're doing instead is just changing where they occur in relation to each other. So typically, if you are the carrier of a reciprocal translocation, you do not show phenotypic effects, so you are phenotypically normal. Sort of similarly to what we saw with our inversions, the offspring of an individual who has a translocation is much more likely to have some sort of significant phenotypic variation. So these are listed here are sort of two mechanisms for how a translocation occurs. These are going to be very rare. One option or one potential is that two non-homologous chromosomes had double-stranded DNA breaks, so essentially a whole piece sort of fell off. During the repair phase, our repair mechanisms then accidentally line them up on the wrong chromosomes. And so again, this is going to be very rare. Usually if you have a double-stranded DNA break inside of a cell, that cell goes through apoptosis because it's less risky to sacrifice the cell than try and put DNA back together and potentially end up with some really weird mutation. This sort of thing is most likely to happen if you have a mutagen, so like a chemical, that is shearing DNA in a cell. Okay, so this isn't usually a natural process. It'd be pretty unlikely for you to just randomly break ends off of chromosomes. So this isn't likely to happen. We can also get crossing over between non-homologous chromosomes, right? So here is an example where chromosome 1 and 7 are lining up in a tetrad. Not usual, not likely to happen, but there are those weird circumstances where DNA base pairs just match just enough that they will do that, and then you get crossing over between chromosomes that should not be crossing over. So there is a specific type of translocation that I want to talk about, and I want to talk about this one specifically because we do see it fairly commonly, and it is a primary cause of Down syndrome. So Down syndrome as a disorder is going to be having 3 copies of chromosome 21. It is essentially the only example of an autosomal trisomy, so an autosomal chromosome in which we have an extra copy, where an individual can survive with a fairly normal lifespan. Okay. What is it about chromosome 21 that makes it easier to survive with 3 copies than chromosome 1? Okay.
It's smaller,
It's smaller.
so it holds less genes.
Yep, so it has fewer genes. The genes that it does contain are non-fatal if we have 3 copies of them. The other chromosomes, if we have 3 copies of those genes, first of all, we have 3 copies of way more genes, and some of those genes are going to be lethal when we have more than 2 copies. Okay. All right, so this type of Down syndrome is called familial Down syndrome, and it's the result of a Robertsonian translocation. So in a Robertsonian translocation, what we have is 2 non-homologous chromosomes. In this case, it's chromosome 14 and chromosome 21. These 2 chromosomes undergo a reciprocal translocation. Actually, first, let me back up. These 2 chromosomes, when we look at them structurally, what are they? Metacentric, telocentric, acrocentric, or submetacentric? here, sort of that waist. Is it in the middle? No, so they're not metacentric. Is it at the very, very end? Yes. Not quite. Okay, so this is an acrocentric chromosome. Okay, so the chromosome, or the centromere isn't in the middle, but it's also not the very tip. So these are two acrocentric chromosomes. The petite arms end up sort of lining up with each other. Both of them share. The two long arms, right, so this is one long arm and this is the other long arm, end up joining and forming one really big metacentric chromosome. And then we get one very short chromosome. This chromosome, sort of the short remainder of this chromosome, is usually lost because it doesn't contain enough genetic information to be useful or necessary in a cell. So what ends up happening is individuals with this type of translocation, again, are phenotypically normal because they have all of their chromosome 21 genes and all of their chromosome 14 genes. But when they go through meiosis, weird things end up happening in their gametes. Okay, so here's an individual that is a carrier of this Robertsonian translocation. So they have a normal copy of chromosome 14 and a normal copy of chromosome 21, but then they have this long metacentric chromosome that contains basically all of the information from 21 and all of the information from 14 and 1. So phenotypically normal, because they have 2 copies of 14 and 2 copies of 21. But when they go through meiosis, they end up with these potential combinations of chromosomes in gametes. So normal, mostly normal, because it's a copy of 14 and a copy of 21. This one has 2 copies of 21 and 1 copy of 14. Only 14, only 21, and then 2 14s and a piece of 21, or and 21. So they make some gametes that are normal, and if each of these is fertilized with a normal, we'll just say that these are eggs, so it's fertilized with a normal sperm, the results are an individual that has 2 copies of the proper chromosomes, 2 copies of the proper chromosomes, so this person becomes a carrier, but they have 2 copies of 14 and 2 copies of 21, so phenotypically they're normal. And this individual here has 2 copies of 14, but they end up with 3 copies of 21 because they have a 21 here, here, and from the sperm. Here we have, these would be fatal combinations because we have 2 copies of 14 and only 1 copy of 21, 1 of 14 and 2 of 21, and then 2 of each. Okay? So there are some cases where Down syndrome does run in families. That sort of situation is more likely to occur if one of the parents is a carrier of this Robertsonian translocation, right? Because they then, of all of their potential gametes, half of them are viable. Of that half, 1/3 of them results in Down syndrome, 2/3 result in phenotypically normal offspring. Okay. So, and then this is just the sort of the mechanism by which we get that break and translocation. So we have 2 acrocentric chromosomes that broke and exchanged long arms and short arms. So we get one really long metacentric chromosome, which is 14 and 21, and then we get that tiny little piece that is lost. Yeah, so here is a karyotype of somebody that has familial Down syndrome. This looks different from somebody who has what we think of as like standard Down syndrome. With the more common form of Down syndrome, it would be literally 3 copies of 21 right here. This individual has this Robertsonian translocation chromosome right here, so it's 14 plus 21 sort of tacked on the end, and 2 copies of the normal 21.
So they don't have any of the phenotypes for Down syndrome whatsoever?
This individual would, because they have 1, 2, 3 copies of it. But if, like this individual, would not.
Oh, okay.
Okay, so I don't necessarily want you to remember this, but I just wanted to show you why we have issues when we have translocations. When we have translocations, remember normally the carrier is phenotypically normal, but during gamete formation, we're going to end up with a situation where a lot of the gametes are going to have the wrong amount of genetic information. So when an individual with a translocation goes through meiosis, they can't form simple tetrads that then divide evenly into gametes. So this is an example of what those tetrads where we have translocations have to do when they line up along the metaphase plate. Okay, so we get sort of our normal chromosome and a normal chromosome, and they have to form these crosses in order to match up or pair up with their homologous pieces, because their homologous pieces occur on different chromosomes. So during the division process, we end up with, there are a lot of different ways to separate these, and I don't want to go through them, I just wanted to show you that this becomes very challenging, right? Because if we divide here, and each of these goes into the next phase of division, we end up with mostly blue chromosomes in one gamete and mostly red chromosomes in the other. We pull them this way, they're a little bit more even. We can actually pull them this way. It gets really complex. Okay? So an individual who has a translocation, usually about half of the gametes will be phenotypically normal or will be genetically normal. The other half are likely to have too much or too little of a particular chromosome's genetic material. So sometimes individuals who have both inversions and translocations are going to experience reduced fertility rates because, think of it, if you're a male, only half of your sperm then are viable. Or a female, only half of your eggs would be viable. And in a female, you produce 1 egg per month. So if it happens to be the fruits version that's not viable, you have a more challenging time getting pregnant. Okay, are there any questions on duplications, deletions, inversions, translocations? Okay, so now what I want to do is I'm going to define sort of some of our changes in chromosomal number, and then we are going to go through non-disjunction events so that we're very clear on how we end up with extra chromosomes sometimes or too few chromosomes in other cases. So when we're talking about changes in complete chromosome number, they either fall in the euploidy category or the aneuploidy category. Euploidy would be if we gain or lose an entire set of chromosomes. So here is an example of a euploidy event. We start off with an individual who has 4 chromosomes, but they occur in sets of 2, so they are diploid. That would be a fruit fly, for example. Diploid, 4 chromosome. Oh yeah, it even says fruit fly up there. So if we have a duplication event of the entire genome in a fruit fly, and we gain an extra chromosome at every location, we end up with a triploid individual, right? We now have 3 copies of chromosome 1, 3 copies of 2, 3 copies of 3, 3 copies of 4. If we double that again, we end up with a tetraploid, so we have 4, 1, 4, 2, 4, 3, 4. Okay, so every chromosome is duplicated. In an aneuploidy, we do gain or lose chromosomes, but it's an individual chromosome. It's not the entire set. So here is an example. Again, we start with our normal fruit fly who is diploid. So she is 2N. When we accidentally copy an extra chromosome, or we end up with an extra chromosome in a gamete, we call that an aneuploidy. In this case, it's a trisomy. So we have 2N still, because we have 2 copies of 1, 2 copies of 3, 2 copies of 4. We have an extra copy of 2, so we say we're 2N plus 1. If we happen to lose a chromosome, it's a monosomy. Again, we're still 2N because we should and do have 2 chromosomes at each location, with the exception of this one. So we say we're 2N minus 1. Okay. So again, an individual who has Down syndrome would be 2N plus 1 with a trisomy at chromosome 21, sorry, chromosome 21. We don't even have 23. We do have 23 chromosomes. We don't have a chromosome labeled number 23. It's almost fall. Okay. And so when we're talking about aneuploidy, this is where we're going to focus right now. If we have what's called a nullisomy, that would mean that we have no copies of a particular chromosome. I can't think of a single example in humans where that is viable. You have to have a sex chromosome, and you have to have at least, well, you have to have 2 copies of every one of our chromosomes. If you have a monosomy, it means that you've lost 1 chromosome. So in humans, a good example of that would be if you had only 1 X chromosome and no Y chromosome. Trisomy would be an additional chromosome, but again, only 1. And so our examples in humans are 13, 18, 21 can exist in trisomies, as well as XXX and XXY or XYY. You can gain an extra sex chromosome, or you can gain an extra, it's not really gaining because it's not like you bank it, but you can accidentally end up with an extra chromosome. Unintentional lack of separation during meiosis. And then tetrasomy would be if you have 2 homologous chromosomes. And again, in humans, I think the only examples would be XXYY or XXXX. Our sex chromosomes are pretty fluid in terms of how many we can have and still be viable. Our other chromosomes are very static. We can't really gain or lose them without significant effects. Okay, we're going to move ahead to our aneuploidy mechanism. And I'm actually we're going to draw this out repeatedly. I know, I've been told how much you guys love drawing meiosis. It's everybody's favorite mechanism. Okay, so we're actually going to, we have 2 different mechanisms for aneuploidies. They both occur during meiosis, and so the first one is a non-disjunction event in meiosis 1. Non-disjunction means that our chromosomes don't separate properly, right? They don't disjoin from each other. So it can happen during meiosis 1, which would be that initial division, or it can happen during meiosis 2. The outcomes are similar, but there are sort of 2 big distinctions, and so I'm going to point those out now. One of the major distinctions in the resulting gametes is going to be whether they contain 2 copies of sister chromatids or 2 copies of homologous chromosomes. Okay, so we're going to walk through this again, but I just want to point that out now. The other thing that's different is if a non-disjunction event happens during meiosis 1, we end up with we end up with 50% normal gametes. No, that's not right. We end up with no normal gametes. Yeah. This one is going to end up with 3 copies of this chromosome. This one is going to end up with 1. Okay? So in meiosis 1, when we have an error, none of the resulting gametes are normal. They all have too many or too few chromosomes. If the non-disjunction event takes place in meiosis 2, 50% of the gametes will be normal, 50% will be abnormal. Okay, so let's draw this out. We're going to start in a cell. I'm actually going to use a male sex cell here, and I'm going to use X and Y chromosomes as our example because it's very easy to tell homologous versus sister chromatids when we're dealing with X and Y chromosomes. So we're going to start with the male. We're just going to look at the sex chromosomes for right now, and I'm going to start in metaphase. So in our first round of division, we are going to end up with 2 X chromosomes on one side and 2 Y chromosomes on the other side. Okay, so remember that even though X and Y are not homologous chromosomes, they line up during meiosis as if they were. We have those pseudo-autosomal regions that helps them line up. All right, so if the error occurs in meiosis 1, all 4 of these chromosomes are going to go into one of the daughter cells. Okay, so our our homologous chromosomes do not separate. Right? So remember during meiosis, during the first division, homologous chromosomes separate. During the second division, sister chromatids separate. So if there's an error in meiosis 1, in our daughter cells, we are going to end up with one daughter cell that has all 4 of those homologous chromosomes. Okay, so it's going to have 2 X's and 2 Y's. The other cell gets nothing from this tetrad. Good so far? Okay, so meiosis 1 non-disjunction is failure to separate homologous chromosomes during the first division. So, in our next round, right, this cell is going to keep going because it has other chromosomes in there which are presumably normal. So this cell is going to divide into a sperm cell that has no sex chromosome. Or 2 sperm cells with no sex chromosomes. This guy up here is going to divide. When it divides this time, the sister chromatids are going to line up as normal. And so in one cell, you'll get an X and a Y, and in the other cell, you're going to get an X and a Y. So we're going to make these sperm cells. Okay. So when these sperm cells fertilize a normal egg, what ends up happening is these 2 are going to produce a gamete that has 3 sex chromosomes, right? So it's 2n plus 1. These 2 are going to end up forming that because they don't contribute any sex chromosomes. So we are 2n minus 1. Okay. And the way that we can tell this particular non-disjunction event happened in meiosis 1 is because we end up with 2 homologous chromosomes, well, homologs, X and Y aren't actually homologs, but 2 of the homologous individuals in a cell. So let's look at this over here. In meiosis 2, so we can see the difference. Okay, so we're going to start with the same cell. We have a sperm cell, we have 2 X chromosomes and 2 Y chromosomes because that's how it starts. This time, meiosis 1 is going to happen normally. Okay, so our homologous chromosomes are going to separate, and we'll end up with our X chromosomes in one and our Y chromosomes in another. Okay, so our sister chromatids end up in different cells, as expected. This time, in meiosis 2, we're going to have an error, and so this time I'm going to say the error is going to occur in the Y chromosomes. Okay. So the X chromosomes are going to separate normally. which means that we produce 2 sperm cells that each have one X chromosome. We are also going to produce 2 additional cells. We're going to say there's an error in separating the sister chromatids. So one of them ends up with 2 Ys and the other one ends up with none, because those sister chromatids did not separate properly. So again, we want to sort of look at the phenotypic outcome of offspring that are made by these sperm. Up here, the egg is going to have an X, and each of these sperm would contribute an X, so we are normal, just 2N. Still standing. All right, the other one, this top sperm is going to make this individual, and this bottom sperm would make that individual. So one of these, this guy has 2 Y chromosomes, so this is going to end up being 2N plus 1. This one doesn't have any sex chromosomes, so this offspring ends up being 2N minus 1. Okay? So, in meiosis 1 errors, none of those offspring are phenotypically normal. Everybody's missing a chromosome or has an extra chromosome. In these offspring, half of them are normal, half of them are abnormal. Okay? Okay, so what I want to do now is, I'm actually going to, I'm just going to run through a few things on these slides, and then I'm going to give you another sort of practice prompt and have everybody write it out on their own, and I'll walk around while you're drawing meiosis 1 errors and meiosis 2 errors. So I mentioned that we do have a couple of trisomies that are non-lethal in humans. So they are 13, 18, and 21. Both trisomy 13 and trisomy 18, when we say that they're non-lethal, we mean that an individual with these chromosomes, an extra one of each of these chromosomes, can be born. They usually have a life expectancy of less than a year. So these are more detrimental than trisomy 21 or Down syndrome, which now has a fairly normal lifespan associated with it. The sex chromosomes act quite a bit differently. Why do the sex chromosomes act differently than the autosomes? What's different about how our cells handle sex chromosomes compared to how they handle autosomes? Yeah, that's going to be a big part of it. So one thing is we have X inactivation. So even if you have 6 X chromosomes, 5 of them are inactivated at the point of fetal development. Okay, so we only express 1 most of the time. What about the Y chromosome? Do you know about the Y chromosome? It's very small. It doesn't have a lot of genes. The genes that it has are obviously not necessary for life because no female has an X or a Y chromosome. Okay, so having more than 1 Y chromosome is also not fatal. Okay, even though we have X inactivation, individuals who have too many or too few X chromosomes still do have some phenotypic effects. Oftentimes, again, these are associated with like lack of fertility and secondary sex characteristics. Because if you have 3 X chromosomes, that's really hard to divide evenly into gametes, so you end up with gametes that have extra or too few, and it creates other issues. So Down syndrome, sort of our traditional example of Down syndrome is going to be a failure of chromosome 21 to properly segregate, and so we'll end up with an error in meiosis 1 or meiosis 2, which ends up giving us some gametes that have too few copies of chromosome 21 and others having too many copies of chromosome 1. You may or may not be aware that odds of Down syndrome increase with maternal age. So as a female ages, the chances of having a baby with Down syndrome also increases. Usually around that 35 mark is where those odds increase pretty significantly. There are errors in sperm production as well that can result in Down syndrome, but they're not as common. So what is the difference between how eggs are made and how sperm are made that would account for a difference in chromosomal abnormalities in meiosis?
Eggs don't go through meiosis two until they've been so when they get released to travel down the fallopian tube.
Yes.
But sperm, they're automatically going through both.
Right, so sperm, the big difference is that egg and sperm are stored differently. So eggs are stored, sorry, I'm really struggling right now. After meiosis 1, but before meiosis 2, right? So at birth, they stop going through the meiosis process, and then they stay arrested in the middle of meiosis from birth until actually fertilization is when meiosis 2 actually happens. So that's a long time for them to sort of be paused, and it increases the rates of error because they're sort of in this weird suspension state, and things get old and don't function as properly. Sperm are always made from new stem cells, and they're made kind of consistently, right? So they're not held in this weird paused state for so long. So this is just the odds of basically non-disjunction events in females as they get older. So very young ages, the odds of having, and this is again this is not familial Down syndrome. So familial Down syndrome would be if you're a carrier of that Robertsonian translocation, your odds are going to be consistent in that case. But here we're talking about non-disjunction events that result in too many or too few copies of chromosomes. And so that increases. 35 is where it becomes basically 1 in 300, and then by 45 it's 1 in 32, 50, good lord, that sounds painful, is 1 in 12. The oldest natural conception in humans, I think, is 55. But can you imagine, at that point, most people are 10 years past their fertility window, and then all of a sudden, hey, surprise. Okay. Okay, so we have 10 minutes left, which I think is a good amount of time to sort of walk through non-disjunction events in meiosis 1 and meiosis 2. I will ask you to just, okay, so I'm going to give you a situation. Let me see what I have here to work with. Okay. So I'm going to have you start. I'm going to give you 2 chromosomes this time. So I'm going to give you the sex chromosome, so we'll leave it here for the sex chromosomes, but I'm also going to give you, actually I shouldn't erase that because you're going to be looking at it. You're going to start here, but then you're also going to get a set of chromosome 21, and I'm going to have you distinguish between the homologous chromosomes by just calling them 21A and 21B. I'm going to tell you that for your meiosis 1 error, you get non-disjunction of number 21. Okay? So in this case, your sex chromosomes will separate normally, but you'll get non-disjunction in chromosome 21. For your meiosis 2 error, show me what it would look like if number 21B did not separate properly. So non-disjunction in 21B. Okay? Okay. So just draw out both of those. Start with meiosis 1, and I'll give you a few seconds head start because I'm going to come around. Well, I can press another one. This is my answer key, so I'm probably I would consider it, but there's only 12 questions this time, so.